Hi, my name is Diana Berrent. I'm the founder of Survivor Corps, and welcome to our webinar series. We know that you can't always get to the experts, so Survivor Corps is here to bring the experts to you. Today, we're going to be talking about plasma, the truth, the myths, the real facts. Let's get to it. We have Dr. Casadevall, MD, PhD. He's a microbiologist and immunologist working with a world-renowned team that looks at diseases and how they impact humans at Johns Hopkins Medical Center. We have Dr. Michael Joyner, who is an American anesthesiologist and physiologist who researches exercise physiology and spearheaded the early action protocol that got plasma into the bodies of tens of thousands of COVID patients to help them recover from the disease. Both of you, sirs, have been deeply involved in plasma, and we want to talk and get to the root of the matter. So, Arturo, tell me about the history of plasma.
So plasma is being used in both the new form and the old form. The old form is serum. Plasma and serum are pretty much the same, except they're prepared somewhat different when it comes to antibodies. But it's been used since at least the first big pandemic of the 20th century, which was the 1918 flu pandemic. It's been used dozens of times usually in epidemic crisis. What happens is that plasma doesn't need any development. It can, once you have survivors, they can donate. So in situations in which you have a crisis with a new infectious agent, plasma has been used, and it's been used repeatedly in prior epidemics, such as the first SARS epidemic. It's been used with the influenza epidemic. Prior to that, Argentine hemorrhagic fever, and many other outbreaks.
And Mike, can you explain how is COVID, what's the history of COVID vis-a-vis, what's the history of plasma vis-a-vis COVID? Well, how has it been used during the COVID crisis? We've been hearing a lot about it. We've been asked to donate it.
Right, Diana. So it's really pretty straightforward. Basically, Arturo wrote an editorial in the Wall Street Journal talking about the past uses of plasma or serum. Arturo is a friend of mine. I read the article, which was in late February of 2020, pinged Arturo via email. We started a dialogue, and the folks at Johns Hopkins had started to develop an early treatment or prophylactic treatment to give it to people to protect them, give them temporary immunization against COVID. And then my colleagues and I at the Mayo Clinic started doing an early treatment protocol. And you and others in Survivor Corps started to work with other physicians who wanted to use it compassionately right away in late March. And then all of a sudden, the Food and Drug Administration is overwhelmed by compassionate use requests. A sort of gray market for plasma springs up. And we got a note from Peter Marks asking us if we would be interested in running an expanded access program for plasma. I thought we'd do a little demonstration project in about 5,000 patients, take the pressure off the FDA for the compassionate use requests that they were getting, which are very time consuming. And really that's what happened in really early April. And then things just exploded as a result of physician interest, patient interest, the blood bankers getting organized, and then people like the folks in Survivor Corps really stepping up and donating. So really, this is a bottom-up sort of a network thing of repurposing old technology to attack a new disease with a lot of community involvement and a number of things that happened to really become almost like a snowball rolling downhill. So what started as a demonstration project really really took off.
So I'll be honest. I was one of those first people to donate plasma. My first plasma donation, I believe, was April 4th. And I was the first person at Columbia University to donate, and I went and I donated 8 times. And then people started asking, like, well, why are you doing this? Is it really working? So, does it work? I mean, was it working over the summer? It was being used on a compassionate care basis. Mike, how'd you get to the point where you realized where it was working, where it wasn't? How did that come about?
Right. Well, really, I think Arturo's initial insights that early was better. And I think this is what he's promoted really throughout, starting with that editorial in the Wall Street Journal, is what has emerged over the summer. And I think what we began to see is that earlier is better, and units of plasma with high levels of antibodies are probably better as well. So I think those were the two key insights, which really, and I think I'll turn it over to Arturo, really amount to us reinventing or rediscovering what would have been common knowledge prior to World War II.
I mean, I agree with Mike. What happened was that as a result of availability, because of donors like you, as a result of the fact that there was no other therapy, as a result of the fact that the FDA has set up the extended access program, use took off. And then the criticisms began. How could you be allowing all this use to be done without clinical trials and without safety and efficacy data? And what Mike and colleagues had here was an enormous, rapidly growing database. So the question was, can you look at it and get some insight? And that's what was done. It's a database where all are treated. So you don't have a negative control, but that's what is expected. The question is, can you look at it and can you find evidence of safety and efficacy? And to their credit, they analyzed that very large database, and they very rapidly established that convalescent plasma was the safest plasma. And then guess what? Looking at the numbers, they saw signals of efficacy. They saw that if you treated early, you were, by that, within 3 days of hospitalization, you were much more likely to survive than if it was treated late. And more important than that, they saw that if people got high titer, they were more likely to survive than if they got low titer. Titer is a word for antibody. So that emerged from that database.
How concentrated the antibodies are in the plasma, is that correct? So what, right? Okay.
Right. So what happened is that that information became available relatively rapidly. In the meantime, people were organizing to do the randomized control trials, but the randomized control trials were getting into trouble because they were being set up in places that the patients in the initial search areas. And then what happened is that as the initial search was controlled, there were no more patients. And then others were being set up, but they didn't necessarily have the insights that came out of the Mike study, that was that you needed to treat early and you needed to use high titer. So what we found is that there was this kind of dissonance in that various forms of inquiry were moving at a different time.
So what happened with early studies?
So I think a couple things is one thing you have to think about the normal way new therapies are developed. You know, there's experimental models, there's early studies in humans, you establish a dose, you then escalate the studies to larger and larger studies and so forth. Well, we kind of started with what would be described as a phase 4 study in the middle of a pandemic, while other things backfilled these other normal steps. So what we have emerging, Diana, is really a mosaic of efficacy. So as we went on this summer, animal studies started to emerge showing that passive antibody transfer, or convalescent plasma, was effective in a number of models. We started getting studies in rare patients who don't make antibodies, showing, or at least case reports showing, that these people did remarkably better when they got plasma. People started looking at treated and untreated patients, not in a formal way, but in a look back or retrospective way, not in a prospective way. And again, started to see these signals of efficacy. So now what's coming online are the results of the randomized controlled trials, and the ones that have used plasma early, prior to the patients becoming very sick and ending up in the ICU, are looking generally pretty positive, where the ones that are done in patients who are sicker and have been in the hospital for a while or in the ICU show maybe less of an effect or no effect of plasma. So a very coherent picture is emerging about early use and the need for high titer antibodies. It's just not the kind of linear chain of studies you have under normal times. And I think that's what people have to recognize.
Right. So you're basically you're learning as you go. But I think, and the other thing that is apparent now is that we have learned a lot about the pathogenesis of COVID-19. So it appears that there is an early phase dominated by virus. And then there is a later phase, the one that gets you into the hospital and gets you really sick, dominated by inflammation. So if you give the antibody relatively early during this virus phase, you essentially prevent the second phase. And that's what we see in some of the early trials. Whereas if you give it after inflammation has set up, antibody is not going to reverse inflammation. The process is gone, you know. This is a runaway train. So that's why the other thing that has emerged is that you can begin to put the findings in the context of what we call the pathogenesis of the disease. And we can understand that the plasma is working as an antiviral. And if you treat early, you treat the virus. And if you treat the virus, you don't get the inflammatory response. And by the way, this has also been confirmed by the monoclonal antibodies. The monoclonal antibodies work early. They don't work late. It turns out that plasma may have a longer window simply because it's a more complicated type of product. It has many different types of antibodies. But what's striking is how all these pieces of evidence are falling into place, but they are not falling into place in the usual linear way that people receive them. Somewhere you may be getting a clinical trial result before you get the other results. And it's just going to take a while to synthesize it all.
So you just put my mind with talking about plasma as an antiviral and sort of breaking it down into these two stages of the viral replication stage and then the inflammatory stage where you're seeing the damage done. And having had COVID, just a mild case of what I refer to as the Tylenol and Gatorade kind of COVID, and still suffering some of the inflammatory effects of that 10.5 months later. The idea of being able to stop that inflammation from ever having started, even in a mild case, would be extraordinary. How early can it be given realistically?
Well, right now, the early use is most notable for the monoclonals because those can be given on an outpatient basis. There are clinical trials, Diana, going on at Hopkins and via the NIH looking at outpatient use of plasma. So, and the main really fantastic study by Lipser and colleagues from Argentina, where they gave plasma to older Argentinians, 65 to 75, people with a lot going on, showed that they were able to reduce hospitalizations dramatically in those patients if they gave them literally plasma in a van near their house. So I think what we have to think about is how we're going to move plasma, monoclonals, or any sort of antibody product as far up front as we possibly can, where it can do the most good, and where frankly antibody therapy historically has been shown to work, whether it was pneumonia in the 1930s, meningitis in the 1920s, and other diseases where antibodies have been shown to work. So I think we've got to just kind of continually push it earlier in treatment. And that requires some logistical doing, and it's a little bit easier sometimes to give things in the hospital. So I think in the coming months, we're going to have to get better logistically at giving these antibody products on an outpatient basis.
So speaking of the administration of these therapeutics, I mean, that seems to be, it's no small matter, you know, that you need to get it as an infusion, because if you have active COVID, how do you get to the infusion center? I mean, who's going to even, who in their right mind would drive you? You know, who's getting
right? And Diana, so the logistics are challenging. The infusion center has to somehow be segregated or separate, or kind of almost a pop-up, because you don't want people going to the infusion center where the cancer patients are and infecting them and this sort of thing. So that's one of the logistical challenges that people have had. Here at Mayo, we've given a very large number of doses, but we have an integrated practice here. Our facilities people were able to rapidly construct a place and remodel a place for the COVID patients to go to get the monoclonals and so forth. So I think like what we're seeing with a whole lot of things, and this is where Survivor Corps has been involved as well, is while you can make tremendous conceptual progress and have technical solutions to COVID or technical things that can help with COVID, there's so much last mile sort of retail elements, whether it's getting people to the infusion centers, getting people to the blood banks to get their plasma collected, getting the vaccines actually in people's arms. So much of what's happening here has become a retail challenge, and is not, as they say, rocket science, but it's just kind of bread and butter, last mile sorts of things.
Speaking of that, where's plasma going now that there's a now that we have monoclonal antibodies? I know monoclonal antibodies cost more, but plasma is tough because as we have more people vaccinated, we'll have fewer people to supply plasma. Hopefully we'll have fewer people who need it. Do you think that there's a possibility that one of these products will end up being an intramuscular and actually keep people from, that they could go in a car, get the shot through the window and just keep on going and never have to enter the hospital? Do you think that's possible?
I think with the with what they call the IVIG, which is a concentrated formulation, they begin to get there, but it's still not what it needs to be much more concentrated to be given intramuscularly, because you can only give a small volume. So the answer is the technology is there, it's possible to do, but the formulations are not there yet.
Oh, that's exciting.
But you ask a really important question: where is plasma going? Well, antibodies are licensed for approved for the outside as an outpatient. Plasma has a role as an inpatient. And I think that even though fortunately the curve is coming down, I think we still have some time in which plasma is going to be used in the hospital. However, there is the big threat in the horizon are the variants. The variants that could potentially derail things. If you have a spread of a vaccine-resistant variant, you essentially will get it again, whether you've been vaccinated or not. However, anybody who has a variant and recovers is a donor of variant convalescent plasma. And then that can be used to treat the variant. It could also be, hopefully by then, we will have the outpatient data available, and then it may be possible to give plasma as an outpatient, and then we can treat people much earlier. They keep them from ever going to the hospital. So I do think that you're right. I do think that now that we have more products, we're going to have intravenous gamma globulin, we have monoclonals, plasma, and we're going to have fewer, hopefully, patients in the hospital. But I do think plasma will retain a niche for the rest of the year.
And I actually see that niche going on longer, because I look at our leukemia patients, our bone marrow transplant patients on certain medications that limit their ability to respond to vaccines and so forth. And I think that there's going to be a niche use for antibody therapy in COVID, and perhaps in other infectious diseases as well, that's going to continue for some time. And I think as Arturo's been working in this field his whole professional career. I'm really a newbie here, and it's one thing it's always interesting to come to a field sort of later without a lot of preconceived notions. And as I've read the old literature, the old papers, pre-World War II, you really wonder why antibody therapy died out to the extent it did. And you see a potentially bright future for certain forms of antibody therapy in certain patients. We have it now for respiratory syncytial virus in babies. We have it with rabies. We have it a few other places. But I wouldn't be surprised if 5 years from now, when we're perhaps talking about something else, Diana, that we will be talking about antibody therapy in a more general context. And I think antibodies harvested from humans will continue to always at least play some role in antibody therapy more generally.
It seems to be the most nimble response that we have in our toolkit. Would that be correct? I mean, for the vaccine, you need to create the booster. With this, if I know I always get accused of using cooking and food analogies, but I think of this as sort of like artisanal plasma. Am I getting this right? So if we want to against the South African variant and we go to Cape Town and we get plasma from residents of that area, and it'd be the same thing as using local honey to treat allergies, correct?
I mean, we already have some of these variants in the United States. I think that what is needed now is to establish a registry and to basically to get ahead, we should be sequencing more isolates to know what's coming. I mean, we're pretty blind right now to what's happening in the community, and there is a lot of virus transmission out there. But once we identify these, you know the person, well, that person is a potential donor.
Well, and I think the artisanal thing is really an excellent way to look at it, because the other thing that people have to recognize is this is polyclonal, and there's a whole bunch of active ingredients and so forth, and that also, when you think about drug development, monoclonals and others, there could be all sorts of secret sauce in the convalescent plasma itself beyond just the antibodies that attack the virus that we need to learn more about as well. So I think the artisanal analogy is a good one. And like I've said from the very beginning, plasma harvested from recovered humans at some level is really the first best sort of biological shot on goal. The blood banking infrastructure exists that can harvest it. And if you look at our map, we literally gave plasma in essentially every zip code in the country, and it was collected all over. And there was this just sort of distributed way it was approached. So it didn't require any single piece of infrastructure. It required a lot of people to cooperate together so we could have a coordinated response with the donors and bring this sort of diffuse infrastructure together in really almost like a jazz band with 5,000 members.
Speaking of which, Arturo, I have a question for you. There's a concern. I mean, speaking of the, you collect plasma, and you were talking before about genetically sequencing. So we have a registry, so we know what strains we have. We're not doing much in the way of genetic sequencing right now. I know that there's a concern that plasma isn't a uniform product. And with recent studies, I know that there's been a lot of concerns about the role of autoantibodies in the disease progression. And there's an obvious concern that a naturally derived plasma product could pass along the bad antibodies along with the good ones. How are you figuring that out? How can we trust a naturally derived product like plasma? And how do we know the quality of each unit? How do we know the titer, the neutralizing capabilities, the presence of autoantibodies, et cetera?
So there are a lot of stuff in your question. So let's start with the easy stuff. The easy stuff is the measuring of the antibody to the viral. That is back in when we started, when you donated blood in April 4th. That was rare in the United States. Now it is.
Antibody test on March 28th.
You had it right.
I had David Ho's antibody test at Columbia.
So you were early, because for many months afterwards, it was still, people were donating and people were using plasma without necessarily knowing the titer. That can be measured now, and it can be measured relatively quickly, and that can be done. The other questions you asked, though, are things like the autoantibodies. Well, those have been associated with people who have been very sick. There is no evidence that the plasma collected from people who recover and are well and are donors has any of those bad antibodies on board. Furthermore, the blood industry tends to screen people who are more likely to have those antibodies, and those don't make it into the convalescent plasma supply.
So what exactly are they screening for? What are they looking for? They're looking for the autoantibodies. What do they see now? Mike knows more about this, but for example, women that had multiple children are more likely to have what are called some types of autoantibodies. They're rare, so the majority of convalescent plasma is fine. So one of the ideas is that can still be used. It can still be used to make gamma globulin. So the important thing is that anybody who has a COVID should donate, because even if your plasma cannot go directly into another person, it can go indirectly to help somebody else, because the industry can take out the antibodies and formulate it into gamma globulin.
Yeah, and I think the other thing, Diana, this really highlights again, when we started this, we were really flying blind with a somewhat characterized product with a sort of an unknown dose. And I think one of the things we're going to have to be ready for as we think about what the lessons learned are and what we need to do to be ready for future pandemics is to think about not so much testing in terms of diagnostic testing, which is obviously critically important, but we have to be able to think about testing across an array of use cases for testing, where there's antibody testing in plasma, looking at some of these autoantibodies, the sorts of variant testing that Arturo's talking about. So I think that testing has really, been a challenge across the board. If we'd had better diagnostic testing and knew exactly how many people had COVID and recovered, we could have those people be a little later in the vaccine queue. So we could only be vaccinating, at least for the first month or two, the people that had not had COVID, and we could make that precious vaccine go farther. So I think there's a lot of things like that that are all wrapped up in early development, rapid development of an array of testing methodologies and infrastructure that we'll need to think about in the United States and other places, frankly, as we think about lessons learned from the COVID pandemic.
Yeah, there's certainly the theory that looking at seroprevalency and basing the vaccine on that, if we have a scarcity of a resource and we can get away with giving one dose as a booster to people who've already had a natural immune response, I say go for it. So you guys have been accused of cherry picking your data. The UK's recovery trial showed that more people died after receiving plasma than those who didn't. Mike, you are specifically criticized for running the EAP, which distributed 100,000 doses last summer when we already knew that late administration was unlikely to work. How do you respond to the criticism?
So I think a couple things. One, the recovery trial was stopped for futility. There was no difference between the treated and untreated. And in fact, the folks in the United Kingdom are looking at subsets in their data where they may have some signals, and they've started to collect plasma and are looking to do early administration. Just heard a presentation by Dr. David Roberts today from the United Kingdom on that topic. I think one of the things that people forget about the expanded access program that you discussed is this started as a demonstration project to take the compassionate use individual IND, investigational drug request off, take that pressure off the Food and Drug Administration. We were going to do 5,000 people and so forth. And then for the reasons we've discussed earlier, things really took off. But I think you also, if you look at when people got treated, there was a lot of salvage therapy early in March or late March, April, May. But as the pandemic continued and the summer went on, people started to get it earlier and earlier and earlier. And they started to get it, on average 10, 12 days after diagnosis. By the middle of June, early July, people were getting it on average of 4 days after diagnosis. So I think the use case and the things that are emerging, we started to look at. In terms of cherry picking our data, I don't think it's a question of cherry picking the data. It's so much of looking at the strengths and limitations of each study and in comparing them to the kind of classical principles of antibody therapy, which are heavily weighted toward early use. So I don't think it's a question of cherry picking our data and saying this study's good, this study's bad. I think it's saying that this study has these limitations, the second study has those limitations, and again, trying to develop a gestalt, a picture of what the overall story is, as opposed to saying one thing is definitive or another. And then also looking and seeing how the monoclonal data seems to reinforce what the plasma data is showing.
Right, certainly nobody is arguing about early administration of monoclonals. Arturo, what would you say to?
I would agree with that completely. I mean, if you look at our writings, we have from the very beginning, the editorial was focused on using it for prevention of disease. What happened is that was very natural. There was no other alternative. Mike ran the EAP. That basically means they collected data. They didn't go around telling people to administer the data, the plasma. What happened in this country was that frontline physicians, having plasma, having no other options, and having a very good theoretical basis for using it, began to use it. And they began to use it in very large amounts. And as Mike pointed out, the early use, was for people who are very sick, and you wanted to do something for them. That's a very natural instinct for physicians. And then as this information begins to accrue, that you need to use it early. For example, the Mount Sinai data appeared relatively early. showing that people treated before the ICU were more likely to do better. And then that has been a consistent theme. But we're still getting reports today and publications today that date from those early days. And they will continue to come out, just like the earlier treatment will continue to come out. What I ask everyone is they need to become an educated consumer. You need to ask, when was it given? You need to ask, did they use plasma with sufficient antibody? Those are important things. If those things happen and you get no effect, then I think the conclusion would be that plasma has no effect. But if the plasma is not used in an optimal fashion, you cannot be accused of cherry picking when you basically look at the data and you say, well, this data could be optimized by early use or use with more antibody.
I think the other thing people have to recognize is this is an expanded access program, and these programs were started in part in the HIV era when they were trying to get promising therapies to people sooner rather than later, and recognize not everybody could be in a trial or everybody was getting cared for at an academic medical center. And again, over 2,700 hospitals participated in the expanded access program. The vast majority of these are not places where you're typically doing randomized controlled trials and are sophisticated university or academic medical centers. So that's the other thing I think that needs to be thought of, is that plasma, for all its many strengths and a few weaknesses, was made available all over the country. And it made available quickly, and we continue to see lots of use now. I think about 25,000 units a week are currently being used in this surge, and the blood collecting community and others have done a great job. And what we're hearing from the field is that in general, people are trying to give it as early as possible. So the fact that we find efficacy and multiple signals of efficacy in this sort of chaotic situation, and that we're able to extract these signals of efficacy in this chaotic situation, to me, is another way to say things look pretty promising. We have to wait till all the data's in and we have all the use cases evaluated. But the fact that we're seeing signals in such a chaotic and diffuse kind of build the plane while you're flying it situation, I think is pretty promising. And many other things that people now generally think are effective in COVID, like proning and other things, have never been subject to formal trials. These were developed based on the experiences of skilled clinicians making simple observations and doing things that they felt were biologically plausible. So again, people are going to have to try to innovate that way in the middle of a pandemic.
Arturo, you said something the other day that really stuck in my mind about how there would be no study of the cosmos if we relied only on randomized controlled trials. I've repeated that several times and I can't get that out of my head. Let's talk about, so the EAP ended when the emergency use authorization came out. Let's talk about politics, sort of the third rail here. How does it help and how does it hurt plasma? Do you think that the EUA was premature? Certainly its reputation took a nosedive in the aftermath of the EUA.
A couple of interesting things there. One, and there was actually a billing code for plasma that came out at roughly August 1st. So the people in the United States government, the technocrats, the people that are responsible for these sorts of things, had seen the evidence and they had started to work on it. How this did or did not get caught up in the geopolitics of COVID treatment is a separate issue. But certainly we began to make the case to colleagues in the United States government that the data were looking pretty good by the middle of July. And I think they did an adequate deliberation and discussed it amongst themselves, different parts of Health and Human Services. And some people are going to be early adopters. Some people are going to be later adopters. And I think it sort of got caught up in there. And then I think, unfortunately, Dr. Hahn, who was commissioner of the FDA at the time, made a very common mistake. He confused absolute risk reduction with relative risk reduction in some comments and was taken to task for them. And he said there was a 30+% risk reduction when that was implied to me absolute risk, where in fact it was relative risk. And those numbers, if you frame them in the context of relative risk, are probably pretty reasonable. So, but we don't have any control over that. And I think that's one of the things Arturo's done, the donors have done, the people in all over the country that are using plasma and treating COVID patients is people have really learned to focus on what they have control over, Diana. And we don't have control over the political dialogue. We have control over the people we collaborate with, trying to make it as easy as possible for people, educating physicians as best we can, and working with people like Survivor Corps on educating the general public, the blood collectors and others, to make sure there's an adequate supply.
Can you just explain for a second, to go back to that statement that Stephen Hahn said about the 35%? My little mind that hasn't taken math or science
Right.
didn't really wrap around what you just said. Can you explain the difference
Right.
between that and what?
So if the risk of death from a disease was 70%, and you did a treatment that reduced it to 35%, that would be a 35% reduction in absolute risk.
Mm-hmm.
If you had a disease that had a death rate of 15% and you reduced it to 10%, that would be a 33% relative risk reduction. So that's all it really is. And it just goes back literally to how people think about things like batting averages and very simple statistics. So you can see how in one you had the 15 to 10%, you have a 5% absolute risk reduction, but about a third relative risk reductions. So it's just simple confusion there. He corrected it as soon as it became apparent to him and so forth. So I think he did his best to correct the problem after the statement. And again, I certainly have made mistakes about absolute and relative risk in my life and conflated the two. So I think, but the only thing is, no, I didn't do it in front of a camera. So I think we have to be a little bit generous in our evaluation of that, and just say it was just a simple misstatement, a simple mistake.
What if one of your loved ones got sick with COVID right now? Would you send them for monoclonals? Would you get them plasma? At what point would you give them each of these things? How would you handle it?
Well, I've had my people I work with are calling me all the time about relatives, friends, and so forth. And if people are outpatients, I tell them to go get the monoclonals. If people are inpatients, ask for plasma as soon as you can get it.
Now, we're obviously reliant on the public to donate their plasma, to just like we're relying on people to roll up their sleeves and get their vaccines. We're reliant on, the only way to get plasma, so far as I know, is from the human body, correct? So what is your pitch? I did it 8 times in my limited time that I was allowed. I had 12 weeks to get my 8 donations in. My first donation went towards the initial Columbia study, and my 7 subsequent donations all went to patient transfusion because I'm AB positive. In plasma terms, that means I'm a universal donor. Before everyone starts correcting me, blood and plasma matches are different. That much I've learned. And to me, it was one of the most extraordinary experiences of my life. Unlike the two of you, I tapped out my science career in high school and never thought that I would ever be responsible for saving another human life. So for me, it was truly one of the most gratifying experiences I've ever had. And I say that really quite seriously. It's an incredible thing to be in a global moment of collective crisis and to actually be able to step in and help without a white coat and without the medical degree, to be able to save multiple lives in a 28-minute session. I mean, it's a pretty heady thing, even just thinking back on it. It's amazing. I highly recommend that everybody who is able to do it. It is your civic duty. It is your moral duty. It is absolutely, there's just no question about it. We are in a crisis and we need to be the helpers. If you had COVID and you are lucky enough that you got through it to the other side, it is the best thing that you can do to fight this disease, as to sign up for as many trials as possible. I I've signed up for about a million of them myself. And go donate plasma. Go to survivorcorps.com. We list all the trials there. We list all the places where you can sign up to donate plasma, where you can receive monoclonal antibodies if you've just recently gotten a diagnosis, FAQs about plasma, about monoclonals, about you name it, it's all there. Let's have some closing words, arguments. Wrap it up. Let's do Arturo. You want to go first?
Yeah, I mean, first of all, thank you for all you've done on bringing in people. Look, we have to go with the evidence we have each day, and we have to evaluate it. And today, most of the evidence is that early use is helping people. And obviously, we will change our mind if additional evidence is produced. But with the data today, it is important to keep up donations. It is important that physicians know to use it early. And more important, it's important that the public seeks help early when they're sick. It could make a huge difference, whether you're treated, on day 7, on day 5, on day 9. So I commend you on all you have done. I commend Survivor Corps. This has been a grand grassroots movement. And until we know otherwise, the data suggests that plasma has saved a lot of lives.
And Diana, I'd just like to add to what Arturo said, and just your prior comments about your experiences. I think one of the real positives here too, beyond the life-saving effects of plasma when given early, and some of the things we've seen in patients who don't make their own antibodies, I think the other thing is that people are always in research, human research, clinical trials, clinical research, talking about community engagement, patient engagement, so on and so forth. And I think you and the other donors have really taken that to the next level. And I think one of the silver linings of this very dark cloud is going to be we have new models for patient engagement, patient communication, working with people, and that sort of thing that hopefully we'll be able to learn from, leverage, and live to fight another day with. So I think we've made tremendous strides in many, many ways. And I think one of the things that we mustn't forget is how much we've improved community engagement through this effort.
Absolutely. I think that this has obviously been a horrible scourge on the humanity. There's no question about it. But looking for the silver lining, I do hope that we're looking forward to a renaissance, that maybe this experience is shining a light on some inequities and problems in the system that we can build back better and stronger. And on behalf of Survivor Corps, I believe that we have really redefined citizen science as citizen scientist collaboration. And that's how I look forward to moving ahead, hand in hand with the scientific and medical community, so we can save lives together. So thank you, Dr. Joyner. Thank you, Dr. Casadevall.
Thank you, Diana.
Yeah, thank you, Diana. Totally agree.
Okay, go to survivorcorps.com and sign up to donate your plasma today. Thank you.